So good morning, everybody. Let's see this. Okay, they're chasing me, no? Okay. No, okay. Okay, good, very good. Well, good morning, everybody. Let's go on. So today I will explain a bit more, or I will discuss a bit more the concepts of machine learning applied to neuroscience, in particular to neuronal cultures, which is my specialty. And then I will jump to other things of interest that are neuroengineering, okay? Neuroengineering, and then some medical applications, which people ask about them during these days. So, so in the last day, we ended like that, okay, having a bit of fun with a 20-euro bill and this painting, okay? That basically I wanted to illustrate here that we don't understand how neurons code information, and that apparently this coding depends on our previous experience, so there is no really a recipe, okay, to understand how the activity of neurons translates into an image or something, yeah? That's very difficult. So there is a lot of people working on that, and that's beautiful because this means that the problem is difficult, so we love it because it's difficult. We are scientists, we like difficult things, right? And yeah, but in this context, of course, people try to develop different tools, different ideas, and we discussed a little bit very quickly the other day about machine learning and artificial intelligence. With Hilda, we were discussing right now that it's a very complicated topic, and remember that conceptually, we have an input that goes into a network that we have here. This is a network, and then that gives an output, right? And conceptually, machine learning, artificial intelligence, these concepts, what you do is that you tune the weights of the connections that you have in your system. You keep tuning them again and again until the output, or the output, is somehow a function of the input. Yeah. So basically, for similar inputs, you get the same output. Okay. Conceptually, this is the idea. But of course, when you go into applications, that is extremely complicated because the input can be a static image or can be a continuous series of data. This network can be very different kinds of network. And I will discuss that later, okay? But conceptually, you have to understand that this thing is very, very complicated. So I think all of us are using MATLAB packages or Python packages that apply artificial intelligence, and sometimes it's a black box. And I agree with Hilda completely that this black box is very annoying. As a scientist, I would like to understand what the hell is going on. And yeah, but we need time. And I agree with Hilda that maybe we should organize a workshop, right? From the very basics to understand how these things work, right? Anyway, so in the quest to work with these problems, remember that I discussed a lot that today people is using machines, thousands of CPUs or GPUs to carry out these tasks with a number of neurons that approach those in the brain. Yeah, this is what Google and Microsoft are doing. And that is a huge amount of resources. Yeah? And in any case, discussing with Christina, we said, okay, that's very cool. It works, perfect. But that doesn't help us to understand the brain. Correct, Christina? And that is a bit annoying. Yeah. So people were thinking, okay, I like machine learning. I like the concept that is brain-inspired. So can we do something beyond that that also help us in neuroscience? And yes, so here I will provide some interesting ideas that a lot of people in the world is developing to try to bridge the two worlds, okay? And one of them, or the first one, is what is called neuromorphic chips, okay? Which basically are hardware neurons. So basically, you connect the transistors in a chip in such a way that they are very, very close to Fitzgerald neurons, but by hardware. And then you can actually play with them a little bit, so you can decide, you can program a little bit the chip, so you can decide which neuron is connecting with which other neuron, and then you can run the system. And then conceptually, these chips consume about 1,000 less energy than a supercomputer in Google or Microsoft. And you see, the current technology is that Intel fabricated one of these chips that already contains 1 million neurons in it, which is basically almost like the brain of a bee. So you start having let's say, a system that is really interesting for us in the context of neuroscience. And the good thing is that, remember, these are hardware neurons. So that the system is extremely, extremely fast and works very well. And of course, the good thing is that then you can program these chips and then implement characteristics like noise or spontaneous activity. Yeah? If the system has noise and the connectivity is the good one, then the system is firing by itself, yeah? Which is a property that normally these big artificial intelligence concepts or applications do not use. So in this direction, I'm working with my colleagues, some colleagues at the university, at the Polytechnical University of Catalonia, the same one as Cristina, where we are actually developing this kind of chips. This is the chip that we have so far. And the cool thing we want to do with it is first verify that, of course, it works. Yeah, these are spiking neurons in this chip. And then what we found fascinating is that we can couple this hardware with real neurons. Yeah, so we can see how they talk to one another and if somehow they manage to self-organize. But this is super, super cool. And yeah, so imagine this is, remember that in many neural cultures, this is the typical repertoire of activity for a network that is connected randomly in a flat surface. So it's what we called embedded random graph or random geometric graph. Okay, and you see, remember that we had this on-off behavior. So in the chip, we could also simulate or obtained this on-off behavior with these hardware neurons. Yeah. So this is the first approach. And then what we can do is to keep tuning the architecture of this chip to simulate different designs that we have in the lab. And they see if they behave as experimental neurons. And if that is the case, then we can say, okay, my system, my chip, looks like a biological network. Let's try to take advantage of that. Anyway, so one of the, yeah. So in this direction, so what we are doing is that we have this chip, and we can actually couple the chip with a neuronal network, the living neuronal network. So the output of this culture can be the input of this one and the other way around. And we can try to investigate if they manage to synchronize, for instance, the two systems, right? Or if I don't know, if I inject some interesting activity patterns in one corner that guy processes and gives feedback to the other, maybe there is something interesting there going on, okay? But let's be patient. Maybe in another talk, I will explain you more the kind of results that we are obtaining, but I find it very, very fascinating. And also remember that, yeah, in relation to the talk from 2 days ago, that I explained that to identify the neural code is so complicated because it depends on our experience. So we encode information, and it's not really, really clear how the hell I can translate spike trains into the concept I'm receiving from my eyes or from my hearing, okay? We don't really understand that yet. And remember that one of the problems is that the biological systems have a lot of adaptation, they have varying attention, and sometimes neuronal circuits, biological ones, tend to make a prediction of which will be the next action, okay? And all these things are driven by evolution. We need to survive. Actually, we're here, and for millions of years, our main goal was to survive. So nature created the strategies for that. So this means that things are very complicated, so we cannot really make, let's say, an accurate understanding, yeah, of how neurons encode information. Hopefully, we will manage to do that. So remember, this is just to illustrate how difficult it is. And in this direction, the biohybrid devices are under development to try to bridge electronic circuits, like the neuromorphic chip, with biological circuits. Why? Because for some people that had a disease or a physical damage in which some parts of the body don't work anymore, you can invent implants or other strategies to help that person. For instance, yesterday on the Spanish news, there was a person that got implanted a chip here in the spinal cord. Yeah? So the chip translates or tries to pass the information from the axons that were cut, okay? The axons that were coming from the brain and that were supposed to go to the legs, yeah? Those axons were cut, and the neuromorphic chips try to translate the information, yeah, from the biological device on one side to the biological device on the other side, and try to help the person to walk again. Another example are the implants, in this case for the eyes, for blind people. So in these people, they get some sort of chip, like a CMOS chip in the eye, And of course, it's problematic because you're getting information to an electronic device, yeah, that has to translate that information for the visual cortex to make sense of it. You understand? And that's very, very difficult. So that's why these hybrid devices are super important, because if we manage to make an electronic circuit that interacts with a biological one, and they manage to communicate fluently and self-organize somehow, that is the first step to be sure that the information that this electronic device is sending to the visual cortex is meaningful. Yeah? Okay? So there is a lot of ideas that are developing in this direction, and of course, we also want to develop low-power artificial intelligence with these chips, and also try to make robots more human. I don't know if people like that, but that is also something under development. And then, of course, something that is very unique of the brain and biological systems is that they are fuzzy. Fuzzy in the sense that sometimes, yeah, I mean, sometimes biology just wants to solve problems as fast as possible, and a solution is enough as far as it works, okay? So in this context, fuzzy logic can accommodate for all these different, let's say, point of view that yes, no, maybe good enough or acceptable for now, yeah? So if I'm in nature and a predator is coming to me, so I have to take a decision very, very quickly, yeah? And maybe maybe I could take a tool and try to fight, or maybe if it's acceptable enough at the moment to run away, for instance, right? So you have to take decisions. And you don't really need to take the best decision, which is artificial intelligence in computers try to do. So people is exploring these ideas. And also, biological devices always keep exploring. We have spontaneous activity. You give some alterations in the network changes and keeps exploring, and so on. Yeah. Then they are adaptable. Yeah. If the activity changes suddenly, that is also something interesting to look at. And for me, what is really nice, well, they take advantage of noise and spontaneous activity, and they are resilient to damage. There is something I will discuss a bit later, that if I take a neuronal culture and I take a knife and I cut the neuronal culture, it will keep firing. And conceptually, I have to kill about 80% of the neurons for the neuronal culture to stop firing. But if I do that with an electronic chip, goodbye, okay? Yeah, and then of course, as Christina mentioned a few times, they are extremely efficient in terms of energy consumption. So basically, the idea that I'm developing in the context of a European project and other people in the world is that we take excitatory, inhibitory neurons, astrocytes, and other biological entities, let's say. We design, oops. What is it? Sorry, I lost, now, here. Okay, we design some kind of networks that we find that interesting and representative of the organization of the brain, and we try to see if the system, this biological system that I tailor, is able to carry out tasks such as pattern recognition. Yeah? So I'm doing that in the context of this European project, and also many people in the world. And for instance, maybe you heard about something very nice that people in Australia called Cortical Labs developed recently. And these people, what they did was to couple the neuronal culture, yeah, with a video game. Have you heard about the news? No? One person, okay, very good. So, of course, first, people got very scared. Yeah, that's why you have here this comment here. So, okay, you couple neurons, biological ones, with a game, then they start to talk to one another, the neuronal culture learns to play the game. Oh my God, this is very scary. People is really, really scary. But it's really awesome. So conceptually, what they did was something like that. I mean, you have your neuronal culture. I will simplify it a little bit, okay? You have your neuronal culture, and the neuronal culture is getting a stimulus from the video game, yeah? And then you get an output that goes also to the video game. And then you're getting feedback. You're getting feedback from the video game and the culture, so the culture is learning from failure to play the game, yeah? And this is a video of how it works from the company. And okay, maybe. Yeah, actually, the good thing is that you have to imagine that the neurons are sitting in the system I was providing the other day. You have to imagine that the neurons are actually in this device, okay? They are in a system like that, okay? So the output, okay, goes to the video game. And you see, and at some moment, they will start like now, okay? They are playing, and the culture you see is able to really hit the ball, yeah, and keep playing. So it's really amazing that you can really manage by providing the adequate feedback, okay? You can really manage for the culture to play the game. And actually, people are so excited about that people now is thinking to try to put neuronal cultures on spaceships to other planets to see, yeah, to see if they can improvise solutions for the hardware when it fails. Yeah. I mean, there are many cool things, and in my lab in particular, I'm exploring more or less similar ideas, and this is my neuronal culture in the chip, and I use these neuromorphic chips, yeah? So the actual biological system talks to the electronic one, and I'm trying to investigate if they are able to communicate, self-organized, and maybe do tasks together. But of course, this is exploratory science, okay? But I find it really, really amazing. Okay, and then another example is my little friend. Yeah, this is my little friend. So this is a more sophisticated experiment that we are conducting. This robot has more sensors than this one. And what we do with this robot is that, well, these are ultrasounds, okay? Ultrasounds, so you can get distances, basically. And then this robot also has a sensor for light. So what we're trying to do is that we want the robot to go to places that are brighter than the rest of the environment. So basically, the robot gets signal from the environment, these distances and light, sends that to the biological system, and the biological system is getting more firings, yeah, as darker is the system. Yeah? So if it is very dark, it's firing a lot. This is giving instructions, but there's a lot of firing. And at some moment, that firing is so strong that the culture elicits a burst. Yeah? And that burst, when a burst occurs, then the wheels get instructions to turn, yeah? to avoid the dark place. Yeah? This is what we are trying to do. And it's very difficult. Okay, you have to be aware that it's very, very difficult. And of course, we like very much the toy. It's super cool. But of course, at the moment, just to be sure that we don't spend years trying that, we are doing this idea in silico. So the robot is at the moment talking to a numerical simulation with ICTP neurons and giving back instructions to the robot. You follow the idea? Yeah? And then slowly we are developing ideas to understand what kind of plasticity you have to induce and what kind of feedback. This is very important, okay? What kind of feedback you have to induce to the neuronal network to really do something. So for the robot to really go to a meaningful place. Yeah? Okay? Any questions so far? Oh, yeah. Okay. Yeah, Hilda. Or.
But in this case, there's no learning, right? No learning in this case.
Yeah. For this experiment, yeah.
Yeah, no learning.
Yeah.
So how could you induce learning in a sense?
How we induce learning?
Yeah.
That's a difficult thing.
Yeah.
That's a difficult thing because what we are realizing is that first you have to define an architecture in your network that is like the human brain in the sense that you have modules. So basically, the idea is that, at least the idea that we're working on is that you can define like 4 modules in the system with neurons there, okay? So then this one gets the input, then the input is shared with the rest of the modules like that, and then you take one module as output, okay? But the important thing is that there is feedback. So the modules can communicate to one another, but one region is input, like the visual cortex, the other one is output, like the motor cortex. And the problem is, of course, how do you tune the connections for that to make sense? But conceptually, this is the idea. And just let me show you. Oh.
Yeah, who else? Yeah, you also have a question.
But yeah, just let me show you that these are the simulations, okay? You see now it's firing like crazy, okay? And these simulations, and then the results of that goes back to the robot, okay? At the moment, it's still not working. Yeah.
About those neuronal cultures, do you have to feed them?
Of course. Oh, yeah, sure. Yeah,
take energy from somewhere.
It's true. Yeah, we have to feed them, but you have to feed them more or less every 10 days, approximately. 1 week or 10 days.
How do you feed them?
And it's super cheap. Basically, you have to refresh the medium where they live. And actually, it's a good question because the people of Cortical Labs, the ones that are coupling the culture with the Pong game, they develop a super tiny microincubator for the cultures are also with automatic feeding. So they can keep the culture, let's say, mounted on a robot or something like that for months. Yeah? So in principle, they should be able, technically, they should be able to send cultures to the space. They should be able, okay? But yes, it's true. You have to feed them.
Excuse me, your elements, they're always biological. It's not that you put particular oscillators in your system
No,
of
exactly.
neurons.
They are always ICTP neurons. ICTP or Fitz-Unagomo neurons. Yeah?
Okay.
Okay. I mean, we try to get, at least remember that this is the robot talking to a simulation. So we develop techniques to understand what kind of learning, okay, you have to implement for the system to work. Yeah, and these simulations are with ICTP neurons, okay? But yeah, when we go to the biology, of course, they are real neurons, yeah? Okay. Okay, so these are the ideas that we are developing, yeah? And then let me just go into another example that is super cool as well, and is reservoir computing, because one of the big problems that I was discussing now with Hilda, one of the big problems is that when you do machine learning, artificial intelligence, is that you have to define the network you want to work with. And this network has to be gigantic. Yeah? So for instance, I put here just 3 examples of about 10 that you can find in the literature. So for instance, you can use what is called Hopfield networks, which are extremely recurrent networks. Okay, so there is all the time feedback going on in the network. Then you have feed forward, where you basically propagate information from one part of the network to another. This can be extended to have what is called layered networks, in which the input goes into a first layer that processes very basic, for instance, shapes. Then this layer goes to another one that quantifies how these shapes organize to make simple objects, until finally you can get maybe a complete landscape recognized. Yeah? So depending on the kind of network and the kind of problem that you want to solve, you have to define different kinds of networks. Yeah? So for instance, this is used to find patterns of data in images. You feed the system with millions of images, and you want the system to tell you if you're seeing nature or you're seeing people, for instance, right? These ones, the feed-forward networks, are used to predict when you're writing text in the computer. Maybe you use Gmail nowadays, I guess. So, but Gmail now is trying to predict what you want to say, right? So for that, people is using feed-forward networks, and so on, yeah? And people said, okay, and of course you need a lot of energy and gigantic networks for this to work. But conceptually, as a physicist, okay, I find it interesting, but as a physicist, I thought, that's nice. But let's imagine that you have a system, a relatively simple system, and what I do is to play with networks that as a complex person, let's say, like random graph, small world networks, scale-free networks, modular networks. Let me put those basic networks, and let me see how the system performs. And this was conceptually the concept of reservoir computing, okay? So if what I put here, yeah, so for standard machine learning, you have to adjust the weights for the system, yeah, just to be clear. And for the reservoir computing, what you do is that you tune only the weights between the output and the reservoir. It's relatively simple networks, and the important thing is that you don't touch the connectivity in this system. You provide a network, random graph, for instance, here as an example, and you don't touch those connections. But what you do is you tune the reservoir, the connectivity between the reservoir and the output. Something extremely simple. And because you tune only, let's say, 1%, 10% at most of the connections, it's relatively cheap in the context of energy. This one thing. And the second thing that is very fascinating is that normally, in the context of reservoir computing, you do not input, let's say, static images, like pictures of people, right? You input all the time something that is changing dynamically. So trains of spikes or similar ideas, okay? So, and this is very cool because the system is all the time changing. And basically what you do is that you have your input, your dynamics of some input neurons, some spike trains, then the system is reacting to those inputs, and here, in a given time window, you will get a characteristic patterns of activity. And then you see if those characteristic patterns of activity change when you change the input, for instance. I will put an example very quickly, but please make clear that conceptually, you inject data, inject inputs, all the time changing. You have a network that is fixed in the context of architecture, but of course, conceptually, you can change random graph, small world, scale-free, whatever you like. And then you tune the links between output and reservoir and see what happens. And this is an example. This is actually, these ideas are not super new. They were developed about 15 years ago or so. And there are interesting papers like this one, but conceptually is that you have the inputs like this, can be complicated trains of activity. Then in this case, before training, the system just has an overall activity, okay? This is the activity that you measure in the output, okay? So the activity here is basically looks like random, so you can say that it is a spontaneous activity. And then what you do is that you keep changing the weights, okay? So look that this means that the weights here are not touched. And then here I represent that the weights are started to get modified, so the output starts to get adapted with the dynamics of the reservoir. And you see after some tuning, then you see the system is able to globally, I mean the reservoir and the output globally are able to provide signals that in this case are like oscillators, okay? This is the concept, and what people wanted to do, and sorry, this is for learning, and then once you have completed the learning, so this is the kind of signal that you desire for your system. Once the learning is completed, then you stop tuning the weights. Yeah. So conceptually, what people wanted to do in this experiment, well, this numerical experiment, was to show that you can modify the connectivity and then get activity patterns that you desire. In this case, like a clock, for instance. So, for instance, if you're developing a robot and the robot has to walk in a particular direction and the wheels have to move with a motor, with something going up and down, then this helps you, yeah, to carry out that task. Yeah. So this is one thing, and of course, one of the most beautiful things is that we wanted, me and other colleagues, we wanted to explore this reservoir computing in the laboratory. The idea was to take real neurons and see if real neurons have the capacity, the biological neurons have the capacity to really discriminate different inputs and outputs, and also learn to do particular tasks, in this case, particular output patterns. And this is very cool, because if this works, then you are a step forward in understanding the neuronal code, okay? Why? Because I have a series of inputs, spike trains, I train my system, and if I get the same output all the time, yeah, this means that I'm understanding somehow conceptually what is that group of neurons computing, okay? So we have this thing. So first of all, we tested, of course, everything first in a simple simulation. And my students, what they did was to take a neuronal network that is conceptually the one I use many times, okay, with about 1,000 neurons that are homogeneously distributed on a surface, and they connect basically with the neighbors. Yeah? The random geometric graph that we have all the time. And then they took a group of those neurons or small regions in the culture, the in silico culture, I mean, and they injected, you see, a train of data, which is this one, injected this input or injected the other one. You see, conceptually, the inputs are different. Yeah? And conceptually, the idea is that you inject one input like this, and then you want to see the response of the entire network. And you see the response of the entire network, your reservoir and output, is different, is slightly different, okay, in this case and in that one. Yeah? It's slightly different, but not super different. But the idea is that you keep tuning the system until you can really get different outputs when the input is different, and same output when the inputs are similar. Yeah. So this is an example. This is what we're doing. Okay. And what we have here is that here we have the case in which the two inputs were very similar. Okay. And they deliver almost identical outputs. And here is the contrast, the extreme case in which we really change. You see, this input and this input are very, very different. They are slightly different, but conceptually they provide almost identical activity patterns. And here I really change the behavior of the inputs. So here I get this kind of response, and here I get a complete different one. So conceptually, what I mean is that when I provide the similar inputs to the system, I get the same output. When I apply very different inputs, I get different outputs. This was in the numerical simulations, and here is an example of what I mean with this. Yeah, so we have, oops. Doesn't want to play? Anyway, yeah, okay. This is the input, And this is the kind of output I'm reading in my neuronal network. And then I can classify, which is what I tried to represent over there. I can classify the different trajectories. Yeah, the blue ones are similar inputs and that lead to the same response. And the orange ones are other inputs that are similar between themselves, but they are different from the blue ones. Yeah? So and then you get that the blue and the orange have different trajectories in the system, meaning that you could really separate the two signals. Okay? Okay, and then we went to the experiments, again with the same chip, and then we ran the same. So we took our neuronal cultures that have this kind of activity all the time. Then we injected the signals, different signals, or similar signals, and we look at the response of the network. And in this case, what we observed, and was very interesting, and is that, you see, when we get inputs that are very similar, yeah, so basically I get here two basic responses in my system. This is the similarity in my responses. So basically, when I inject, you see, signals that are very, very, very similar, like this block here, I get similar output. And when I go into a contrasting different input signals from that one, I get another group of responses over there. So basically, similar inputs, same response, different inputs from the previous one, but similar among themselves, lead to a complete family of responses. So basically, this system biologically is able to give me two separate responses, okay, for different signals. Yeah. And we kept exploring this idea, and with my colleagues in Japan, we are developing very basic modular networks like this one, in which we do not have neurons anymore on a homogeneous substrate, but what we do is to put them in this kind of crazy modules that we have over there. But conceptually, it's the same. We define as input these regions in the neural culture. Okay, this is my real neural culture in the lab. Okay, I can define different regions to be the input, then my reservoir and the output. But conceptually, the experiments also work very well. But conceptually, what I want to show you is that when I compare the results of this classification, okay, of input signals, what I get is that the accuracy in representing, in giving me the correct response, the accuracy is better when I have a modular network than when I have a random graph. Yeah? And this works very, very well. So we are super happy in the sense that, okay, reservoir computing seems to work in biological systems, seems to work well. Of course, now I can work much harder to induce training. So the output is not something arbitrary, but a signal that I really want for some reason, okay? But the important thing is that when I go from a random graph to a modular network, the system is capable to carry out this separation of signals in a much better way. And this indicates, of course, for us, this indicates that modularity is a trait that neuronal circuits have, and that is not there by accident, because it has a functionality that in this case helps to better process the information. Yeah. Anyway, more or less clear. More questions? No? Yeah. Sorry. Yeah. I can repeat maybe. Yeah. Sorry.
Yeah, how are the sensitive input from the little car?
Sensitive. You into the car.
Yeah.
Oh.
Please
Oh, okay.
repeat
We
the
have
question.
no, sorry.
Can you
Sorry.
repeat
Yeah.
the question?
Yes. The question is how sensitive are the input signals that we are getting? I mean, imagine that we do this experiment with a car. Yeah. If we do this experiment with a car and the car tries to discriminate different inputs, no, right? How good will be the car coupled with a neuronal system, yeah, to discriminate signals or inputs, yeah, from the sensors, right? This is the question. How easy can we implement reservoir computing using our car over there, right? Well, it's very difficult. It's very difficult. We have not done it yet, yeah. But we believe that it depends on the environment of the car that you have. I mean, you will need a very clean environment to be sure that the system is able to discriminate inputs that are very similar or very different without a lot of noise. Because when we run experiments in the laboratory with people walking around, the robot was getting crazy. So you need you need to do experiments with a very clean environment. Yeah? It's difficult. It's ongoing research, and at some moment we will finish it. Yeah. anyway, so I wanted to finish this part here, and then I wanted to go back to, one moment, I have to stop. Oops. And I wanted to briefly discuss briefly discuss some aspects that people ask me, and I think they're interesting. Yeah, I mean, remember that now I presented neuronal cultures that were prepared either on a Petri dish and they connected randomly to one another, or on a Petri dish and they connected randomly to one another, or in this modular architecture. So basically, I wanted to comment briefly a little bit how we can engineer neuronal cultures in our laboratory and why are they interesting. So this is the repertoire of the cultures that we can prepare. This is the standard homogeneous network. This is the one with modularity. And this is called actually engineered modules because we place neurons in locations that are predefined in the network. And this one is the same concept, but neurons just are randomly grouping to one another in different locations that are totally arbitrary in my system. Yeah? So here I always have 4 modules, and here I have about 50 of these supergroups of neurons, but they are connected randomly to one another. And finally, the fourth design is this one in which we put stripes on my system, so neurons, like that. So neurons have to use those tracks or use those tracks to make connections along a particular direction. So basically, we are breaking totally the symmetry of my homogeneous network. And actually, for you to realize that these things are relatively simple, I brought a sample of this kind of patterns. Yeah, so basically, you can share them. Oops, sorry. So you can touch these things, and basically you will notice that there is a surface over there, yeah. And this conceptually this represents the one at the bottom right, but also we can make designs similar to the one on the top center, and there the neurons grow in these patterns, and they change the connectivity depending on the pattern that we have over there. Okay. Yeah. So it's very you can share. Okay, you can share among you all these patterns, and it's very nice, and it's conceptually it's a very cheap idea, and it works very well. Yeah, and I will show you very quickly some of the examples and why are they useful. So for instance, this is the first network. Remember that when I have neurons growing on this flat surface with nothing else, they have this on-off behavior, and when I play them, in this case with this self-organized modular design, you see they fire in small groups. Okay, so this is very nice because this indicates me that I was able to disrupt this super going synchrony in my system, and I like that very much. And this is cool. This is very cool because in the context of data analysis, that I was insisting all the time that you have to be careful when you analyze the data, this is nice because this gives me a very beautiful way to test that what I see here as a dynamical patterns, characteristic dynamical patterns in my network correspond to functional communities in my analyzed data. Where functional communities, I mean that groups of neurons that tend to talk to one another more often than with the rest of the network. Yeah. So you see, comparing the video in which these groups, for instance, these two fire together quite often, that corresponds to well, this group and also these ones that correspond to the green community, and these ones over here correspond to also to this one over there. Yeah. So this is very nice because this gives me a beautiful example that my tools for analyzing the data are working. Okay, and we'll skip this. And then what we used with this data was to develop a model for stroke, in which what we did was to, using a laser and a very sophisticated microscope and a laser, what we did was to destroy one of these aggregates over here, and then the idea was to investigate how the system was degrading and if possible, see if it was recovering after the damage inflicted by the laser. So conceptually, and because, of course, again, right, because we are physicists, we like to simplify, okay, right. But conceptually, the idea is that we know that the brain is modular, so let's make a modular network, and then let's play with damage in the modular network and see how it behaves. So here is the activity before the damage. You will see that, you will see, it's hard to see, but neurons are firing, okay? And then you will see that once we do the damage, all this region is basically silent, and only this corner is active. Sorry for the quality of the images. The images look very bad because we were recording at very high speed, just to be sure that we could follow the evolution of the network. So this is before damage. And then after damage, you see we destroyed just a single group of neurons over there, but only those here are actually firing. And the system is really having a hard time to activate. Nothing. No, it fired now. Did you see it? Okay. So the system really struggled to activate. And you see we deleted only one of these 60 aggregates, more or less, yeah? So which is really just 2%, 3% of the network was enough to cause this damage in activity. But the amazing thing is that if we wait for some time, then we realize that the network, so this was the beginning, then we destroy this aggregate here, so, and then all these aggregates become silent, and you see the density of connections, functional connections is very low, and slowly, you see in just 18 minutes or half an hour, the system reaches a complete reconnectivity except for the killed aggregate. Yeah? So this means that the system, the biological system, is able to rewire itself. Yeah? So it generates new connections or strengthen the existing connections to be able to go back to the original state with firing with everybody. So this means that the biological systems are extremely resilient to damage, yeah? And they can really recover. Question? Yeah. The time.
Can you follow for more time?
Sorry, yes. So the question was if I can follow these cultures for a long time, and the answer is that yes, I can, yes, exactly. I can record from few minutes to several days or weeks, in principle, yeah. Actually, we do that in other papers. We damage when the cultures are very young, and then we follow the development, the recovery along several weeks, yeah. So yeah, it's possible, yes. Okay, so this is the idea, and then to finish this part, another example, quickly, these are my modular networks, and again, remember that I'm all the time obsessed to try to suppress this ongoing synchronization, okay? So, in this case, when I have these 4 modules, you see these 3 guys fired, then this module alone, then 4, sometimes neurons inside this one of these modules. So conceptually, the idea is that this system is not firing synchronously all the time. Okay, this is the beautiful thing that I wanted to achieve, to demonstrate that as soon as I have modularity and I tune it properly, I can escape from this super synchronous behavior. Okay, yeah. So then I will comment on other things, and well, very quickly, for you to realize how important is the connectivity between these modules, if I put a lot of connections between these single units, what I get is that the system is synchronous all the time. Okay, this is the concept of this triple bond description over there. When I put too many connections, one bridge, and another one, then the system is synchronous all the time. This means that I have to tune a lot the connectivity between the modules to achieve a system that is rich in behavior like this one. Okay, and this is fascinating because this gave us tools for us to understand how much can we play with a modular system to make it dynamically as rich as possible. Okay, I don't want to extend a lot. I will skip this, but just I want to show you that you can scale the system up. You can make a lot of modules. And in principle, you could in principle design architectures between these modules that resemble the macro connectome of the brain, in principle, okay? And then start playing to see how you have to couple these units that conceptually, okay, are brain regions. How do you have to couple them to have an overall dynamics that is approximately the one that you get with fMRI, EEG, or similar things, okay? But these are just toys, okay? Yeah. Oh, sorry. Yeah, it's an image that I was showing towards the beginning. But yeah, that is a beautiful image in colors that you have the brain, and then the major fibers that connect different parts of the brain in different colors. Maybe you have seen these images. But okay, but they are more or less famous. But yes, you can get a good estimate of the macro connectome. Anyway, these things are very beautiful. I will skip that. And just finally, to put an illustration, this is our experiments with the trucks, in which we use a technology that is extremely cheap and versatile, and which that we use actually printed boards, the ones that you use for a radio, okay, or similar systems. This is fiberglass, this is copper, and then you put something called PDMS, which is a polymer on top of it. You cure the polymer, you take it out, and then you get what you have in your hands, okay? And then that polymer, you can punch it, you make holes, and then you have the PDMS. Then you have this thing, you punch it to have a culture of a size that you desire, and then you put the neurons in this topographical pattern. And then you investigate the dynamics. And again, of course, our obsession was to get rid of this behavior again, and then this is what we finally obtained, okay? Oh, come on. Please play. Now, okay? So you can see that sometimes two tracks fire, like now. Sometimes you have very slow waves. Sometimes half the culture fires. Sometimes the entire culture, and so on. Okay. So conceptually, you are getting a very rich repertoire of activity patterns in this network. Okay. And this is very beautiful because conceptually, what happens is that each of these tracks behave as some sort of module. Okay. Some sort of a module similar to the ones that we had here, yeah, but with a different design. But conceptually, the idea is the same. As soon as you have many neurons that connect along the track, then you have like an entity, like a community, and because you have this topographical obstacle that the neurons have to pass, okay, it's very improbable or low probable that neurons on the top of one track connects with neurons on the top of the other track. It's highly improbable, but happens. So you have high connectivity along the tracks and weak connectivity transverse to the tracks. And that's why you achieve this rich repertoire in behavior. Of course, I put just here this graph so you can see that for this case, activity is highly synchronous. There is the raster plot. It's extremely synchronous on-off behavior. And here you have a much, much nicer repertoire of activity. And a bit related to Osvaldo concepts, I can say that this system is very poor in the context of, let's say, complexity, because I get all the time the same repertoire. And this system is much richer in the context of complexity. I have a rich variety of possible spatiotemporal states in my network. Yeah, is that clear? Yes? OK, so I don't want to go far away, very far away. Just let me show you something else that to finish in the last 5 minutes. That people ask me, now we change a bit the topic, OK? So we explained a little bit how you can design in vitro different networks with different properties. The homogeneous one, it's cool, it works, OK, that's fine, it's a nice reference, but it's too simple. We want to escape from this on-off behavior because it's very far from the natural dynamics of the brain. We implement different tools to induce modularity, and that gives us a rich repertoire of activity patterns. So this is nice, but now let's move to something else very quickly, because people ask here during these days, and is what can we do in the context of medicine, and if we are actually developing or you are assisting the medical community to develop new tools. And this is an example of what we are doing. For Parkinson's in particular, there is this technology called induced pluripotent stem cells. And in this technology, what we do, well, I don't do that, but my colleagues, what they do is that we can take cells from the skin, and then you can reprogram those cells from the skin to be pluripotent, and from that you can get any cell that you like, in this case, neurons. And then if you do that with people that has Parkinson or people that is healthy, in principle, you can prepare neuronal cultures in which you have one is healthy and the other one is affected by the disease. Yeah? So basically, it is what we do here. Yeah, this will be the Parkinsonian culture, let's say, and then this will be the control, nothing happens. And then what you can do is that if you know that the disease is caused by a gene, in this case, a genetic mutation, what you can do is that you can use CRISPR-Cas9 to edit that gene, okay, delete it, and then you get what is known as an isogenic cell line, which is the cell line that has been cured for the anomaly. And then you can compare if these two, the control, healthy, and the cured, you can investigate if they behave in the same way. If that is the case, then you have evidences that this genetic treatment is working, yeah, and that you can recover the behavior of the neuronal culture. And actually, we spent so many efforts in these things that we got this award from the city of Barcelona from our efforts in developing therapies that the medical community could use, yeah, to treat real people. But the good thing is that the therapies that we develop and that are published here, these therapies were first tested in vitro. Yeah? We demonstrated that they were working, and then we were convincing the medical community to say, okay, this works, now you can treat real people. Okay? But one of the fascinating things, that's why I wanted to talk about this to finish my lectures, and is that remember that I insisted a lot that we have astrocytes in our system, right? And PD, in one of these papers, actually the one in bold, in this paper we demonstrated that for a particular mutation, genetic mutation, which is this one, for this mutation, the neurons are healthy, but are the astrocytes that have problems. So the astrocytes cannot take care of the neurons, the astrocytes die, and they cannot nurse, they cannot keep properly the health of, in this case, of inhibitory neurons, so the inhibitory neurons indirectly also die. Yeah? And that is a problem because, of course, if after all the talks that we have seen, if you have a system where you have excitation and inhibition, and the system is rich in activity, if you suppress the inhibition, the system gets wild. Okay? It gets wild, super synchronous, right? And this is what happened here. And this is what we basically got in the experiments. So first we did two things. First, we look at the individual activity to really observe that the neurons were dying, is what I tried to illustrate here. This is the healthy neuron, and then you see that it's firing, more or less, okay? And the diseased cell, you see it has a very tiny spike and nothing else. So something is very wrong with this poor cell, okay? And if you look at the video, this is the control, high activity, neurons are firing everywhere. You see the flashes, right? Okay. You see the flashes, and then when I go to the diseased one, you see I see nothing. I know that neurons are alive, but they're dying, okay? And they're unable to fire. But what I wanted to put at the end was that the collective activity is also affected, and in a very weird way, okay? And that is what makes these experiments very difficult. You have neurons that are dying of a particular population, but yeah, so you know that about 10% of your neurons that are dopaminergic will die, but globally, the system is more excitable. Yeah? So it's totally confusing, and for many people, it's somehow a mess, right? So if neurons are dying, why the culture fires more often, more synchronously? Well, because the information about the neurons alone is not enough. You need also to look at the collective behavior. You follow? Yeah. And this is what happened here. This is our control culture, in which you have a little bit of synchrony, but it's not super spectacular. Yeah. And if we put a threshold somehow there, you can quantify something called extreme events. I mean, how many activations occur above a threshold, and you see there is only one in the entire recording. For the PD case, you see a lot of events are over-synchronous. Yeah? Something is very wrong, and you can see in the raster plot that activity is very synchronous. And finally, for the ISOF, the isogenic PD line, you see that it's still more synchronous. The peaks are higher than for the control, yeah, but it's much better than this one. Yeah, so conceptually, the isogenic cell line has been conceptually recovered from the damage that it was suffering. Okay, yeah. Okay, I think it's time to stop. Maybe we'll stop here. Yeah. Any more questions so far? Curiosities? No. So let's stop here. Yeah. Okay, thanks.
Any more questions? No? Okay.
Ah. Jesus.
Very nice lecture. I have a curiosity. When you were talking about reservoir computing and you say, well, we have a random graph, we have an input, and we have an output, and we check that similar inputs gives the same output,
Yeah.
right?
Yes.
But my question is, this is a random graph. If you consider another random graph with same nodes and same connectivity, this input correspond to the same input as in the other or not?
If you change the
If you change the
connectivity,
layout, yeah.
Yeah, it changes.
It changes.
Yeah, it depends. You need a minimum connectivity.
Mm-hmm.
Actually, you need an optimal connectivity. It cannot be neither too low nor too high. You need, otherwise you get
No,
crazy
but
behavior.
keeping the same connectivity.
Oh, that we have not tested that. We have
Okay.
not tested
Because
that.
I don't know if the question about which is the topology that even changing the layout or some rewires that naturally
Yeah.
occurs
What-
preserves the output is something that
Yes.
we can that you can test.
Exactly. What I can tell you is that if we change the topology of the network,
Mhm.
so we go from random graph to random geometric graph to scale-free networks, a small world, when we change that, let's say the capacity of the system to discriminate the signals can be better or worse.
Mhm.
It depends on the topology of your system.
But solo only to discriminate,
Yeah.
not to reproduce.
Yes, exactly.
okay.
Yes. But it's complicated because it depends on the nature of your input as well. So you have to, explore it a little bit. Yes, but it depends on the connectivity. Yes.
So going back to the robot,
Oh, yeah.
um,
Okay.
how many neurons do you currently have in that thing?
In the biological culture?
No, in silico.
In the chip.
Mhm.
For the simulations, for the
Ye-
first part. Okay,
For,
about
for the
1,000.
you're controlling the robot with an in silico
Correct.
culture.
Exactly. At the moment, we are coupling the robot with a numerical simulation, and we have about 1,000 neurons.
Okay, because my question is, like, how does that compare to like traditional reservoir learning? Is like the order of magnitude the same amount of neurons?
Yeah, for the reservoir computing, we are using between 1,000 and 5,000 neurons, but reservoir works already with 100 neurons. Already.
Traditionally.
So we have, so artificially and experimentally.
Okay.
And experimentally, my colleagues in Japan, they showed that if you have only 100 neurons, experimentally, the system is already capable of discriminating signals.
Okay.
Yeah? So 1,000 neurons is more than enough.
Okay.
Simple. Yeah.
Cool.
Thank you.
And in the example that you showed, the neurocultural learning, I'm still thinking why the neuronal culture is learning, because
It's learning.
yeah, because there's like a reward or something like that
Yeah.
involved. Or
Yeah. Actually, that is complicated because it's a very good question, because one of the problems of neuronal cultures is that they like to be in a homeostatic state. So it's very, very difficult to teach them. And you have to teach them, you have to introduce a lot of input signals or to provide feedback, a lot of feedback, and stimulate maybe the same group of neurons all the time to make sure that they do not go to the homeostatic regime. It's very, very, very difficult. But yeah, you can induce learning, but as soon as you stop doing the learning, the neuronal culture will try to go back to the homeostatic state. So conceptually, you have to all the time give inputs and do something to keep the system in the scenario that you like. Yeah,
Really interesting.
It's a mess.
Yeah, makes perfectly thermodynamical sense what you're saying.
Yeah, but also it makes sense in the context of the brain. I mean, as soon as you do not use some particular cortical circuits, they are recycled somehow. Yeah, I think. Yeah.
Excuse me. The
Oh, sorry.
others don't listen.
Is there?
Does that mean that they forget
They
what
forget.
they learn?
Correct. They forget. But I think that. It makes sense because in the brain you have all the time getting stimuli. Yeah, And that stimuli keeps the system going or working in the proper conditions. I mean, to have the cultures totally suppressed from input is wild. I mean, it's getting really, really crazy, yeah? Okay, I can go.
I actually have two questions.
Okay.
to ask for. Firstly, relating this last talk, have you guys considered the aging of the cells?
The age.
The aging.
How old they are, yeah.
How old they are to, induce plasticity and things
Oh,
like that?
it's a good question, yeah. It's a good question. I mean, the problem that we have in these experiments is that we do not have yet which is the optimal day to start training the cultures. I mean, ideally, it should be when they are very young, because then you can really help the system to wire in a way that you like. Because if they are very mature, they have made a big super-connected network, and it's very difficult to delete some of the connections. But it's difficult. It is one of the big dramas for people working experimentally in these problems. You don't know exactly what the best moment to act on the network. Yeah.
But you can consider it to explain the diseases that you're trying to modulate, right?
Oh yeah. They reach some state that you like. Yeah.
Yeah.
Yes. But to keep that state going on, and which is the best time to do it, is very difficult. Yeah. Should we stop? Yeah. Yeah. We stop.
You do it afterwards. Well, Christina needs to make an announcement now. So, thanks.
Okay, thank you. Thank you.
No, today, you have the presentations, about 10 minutes per group, including questions. So try to stay more or less on time. And the other thing is, there's one group that needs to start first because they're leaving. Do they have any other constraint time-wise? Otherwise, we'll go by order. Group 1, group 2, and so on. You know which group you are? And, yeah? So what time do you have to leave? What? What? 3 p.m. Okay, but the rest of the group can present. Right, okay. All right, that's fine then. It's okay. What else? Yes, please send your presentation.